We have a question and answer session scheduled for today, next door in 319. I can be there by about 12:10. I have to meet with a student at noon. That should only take a few minutes. And I can give you till about 12:45. So this needs to be, relatively efficient in terms of, getting all the questions answered. Ideally, you've gone through, you've done your studying, a couple, hopefully minor things that you still need clarified. So hopefully you have that written down and ready to go. So we have kind of a short time. So like I said, it needs to kind of be efficient rather than what I'm finding to be normally the case as I stand up in the front of the room and you guys sit there and do that for 45 minutes. Okay? So, there we are. So if you have questions today around 12:10 or so next door, is the place to be. Okay. We're going to move from our look at the cytoskeleton to, for the most part today, sort of on the outside of the cell. Okay, we're going to look at cell walls in bacteria, fungi, and plants, and then move to the extracellular matrix, which is sort of the equivalent, although not exactly, in animal cells. And then we'll finish up this topic on Friday with junctional complexes, including eukaryotic cells, which will definitely link us back to aspects of the cytoskeleton, although we'll see that today as well. So we're going to start out with cell walls. And we talked about them briefly when we did carbohydrates. When we did polysaccharides, we looked briefly at the plant cell walls, so we'll kind of review that at the end of this. But plants are not the only organisms with cell walls. Bacteria have them. That's where we're going to start. And also fungi. So 3 of the 5 kingdoms have external cell walls, and they all have the same function, which is basically to protect the cell from osmotic lysis, right? That was the other context that we looked at cell walls earlier in the course, right? When we looked at osmosis, you can't keep water from rushing into the cell, so you need some way to cope with that. So during the storm, you can't keep water from filling up your basement, necessarily, but you can take some steps. So we saw some things have sump pumps, right, to pump them out. Animals like us have an isotonic kind of situation. And the third solution is basically a very rigid, tough cell wall so that the cell expands, hits the wall, and therefore doesn't burst. And so the strength of the cell wall is greater than the pressure of the water, so it doesn't end up exploding. So plants, animals, bacteria all solve that problem by creating these cell walls. And the other big commonality that we're going to see is that they're all going to be made up of protein and carbohydrate. And the difference between the 3 types of organisms is going to be specifically what carbohydrates and specifically what proteins. But they're all cell walls are a mixture of those 2 biological molecules. So presumably, I'll start with bacteria, so that's why we're going to start with them. And back in the 1800s, the latter part of the 1800s, the 1880s or so, a scientist by the name of Christian Graham came up with a staining procedure for bacterial cells. And it turned out to be a type of staining procedure that we now call chemical stain because it differentiates 2 broad types of bacteria. So if you run a Gram, so the stain is now named after him, so it's called a Gram stain. If you run a Gram stain on bacteria, you can get one of 2 colors at the end. Cells will either stain purple at the end or pink. So hence differential. And at the time, they just sort of kind of arbitrarily referred to this as either gram-positive, so the purple cells were called gram-positive cells, and the pink cells were called gram-negative cells. So they were able to sort of categorize, I guess, bacteria into these two broad groups, those that stain purple at the end versus those that stain pink. And we've kept this nomenclature, but now we know sort of why this happens. The difference is that bacteria have two different kinds of cell walls that we still refer to as the gram-positive cell wall, which you see here, if you jump ahead a couple slides, and a gram-negative cell wall here. So we now know that the staining difference corresponds to a slightly different setup for the cell wall. They did not at the time, but we know that now. So we're going to look briefly at both types, and we'll start out with the simpler of the two, which is the gram-positive cell wall. So these are the ones that stain purple at the end. So let's see. Things like if you've ever heard or had a staph infection. Staphylococcus is a gram-positive cell wall. If you've had strep throat, streptococcus is a gram-positive cell wall, just as a couple of examples. So this one is the simpler of the two, and if you understand gram-positives, the gram-negatives is just sort of a variation on this. And so this very thick section here, from here to here, is called the peptidoglycan layer, and it tells you what it's made up of. Peptido, protein, glycan, sugar. Okay, so we've already let that cat out of the bag there. So it's going to be made up of protein and sugar, and then we'll come back to these purple and green things a little bit later. So let's start with the sugar part. There are two types of sugars in a gram-positive cell wall, and these are shown here in the little ovals here, the yellow and the red ovals here. So the yellow ones are representing the sugar N-acetylmuramic acid, so it's a derivatized sugar. Well, they both are. And the red ones are N-acetylglucosamine. Luckily, we abbreviate these most of the time. So N-acetylmuramic acid is NAM, and N-acetylglucosamine is NAG. So that makes it a little bit simpler. And as you can see, they alternate. So NAM, NAG, NAM, NAG, or however long that polymer is. So we have these long polymers of alternating sugars. These two sugars are alternating. And of course, they're linked by glycosidic bonds, just like all polysaccharides are. And if you like structures, here are the structures of those two sugars. So there's NAM and there's NAG. So that's basically the sugar part, or these long polymers of these two alternating sugars. But if you just sort of line up parallel lines, and you don't attach them, then you can't really make a solid structure, right? It's like trying to build a building out of sort of steel beams that you just sort of have lying next to each other, right? That doesn't build a solid building. Well, neither would just straight polymers of sugars that you're not attaching. So the protein part comes in by sort of cross-linking or attaching these polymers together. So if you'll notice here, off of the NAMs, the yellow ovals here, are 4 blue circles, and those 4 blue circles are representing 4 amino acids. So this is usually referred to as the peptide tail, or the tetrapeptide tail, since there are 4 of them. So attached to the NAMs is the tetrapeptide tail. So 4 amino acids, and they're shown here as well. So L-alanine, D-glutamic acid, mesodiaminopamelic acid, and D-alanine. At least a couple of odd things here in these 4 amino acids. Anybody give me one or both? One might be more obvious than the other. Oh, you got the less obvious one. Two of them are Ds. We learned way back in, I guess, either late September, or late August or early September, that living things use the L amino acids most of the time. Here's our big exception. And so 2 of the 4 are D amino acids, interestingly, rather than L. And I thought that was the harder one. What's perhaps the more obvious one? It's not so obvious. Is there one perhaps you've never ever heard of or seen before in your whole entire life? All right. This third one is, certainly not one of the 20 we studied back in September, right? So this one's a sort of non-standard amino acid. So we studied the 20 standard amino acids, but there are other amino acids out there. This is one of them, but it's not one of the 20 common amino acids that you find in most proteins. So there are a couple of exceptions here in this tetrapeptide tail. Okay, so that's what's represented here. Okay, so off the NANs is these 4 amino acids, but we still haven't linked them together yet. And that's what the little purple rod here is representing, is the linkage between the tetrapeptide tails. Technically, there are 2 ways to do this. There are sort of 2 different things that this purple rod can represent. The one that's shown here in this slide is that this purple rod can represent penta 5 glycines. So sometimes the cross-link are 5 glycine amino acids. So that's a very common way to do it. So the linkage between them are either 5 glycines, that pentaglycine bridge, some bacteria have that, and others, this purple rod represents just a direct connection, a direct, just simple covalent bond between the amino acids in the 2 different tails. So depending on the specific organism you're looking at, it's usually one or the other. Those are the 2 most common ones. Or simply a single covalent bond. Slightly depends, but those are the 2 big ways of connecting these together. And it's usually, as you can see here, the 3rd amino acid of one tail and the 4th amino acid of the other tail is usually, for both of these, how it's connected. So that gives you sort of the cross-link between these polymers of sugars. So now we've created sort of this now solid structure that is just kind of put on layer after layer after layer to create this relatively thick peptidoglycan layer. Okay, so this is representing just layer after layer of this kind of situation. Right. So that's the bulk of this peptidoglycan layer. There are these layers of these alternating sugars in polymers, and the polymers are cross-linked either by 5 glycines between the tetrapeptide tails or just a simple covalent bond between the tetrapeptide tails. But then they have these other elements here that are drawn here, sort of these purple and green cylinders, and they're very, very similar to each other. They're both something called teichoic acid, which I thought I had a picture of, but I guess I don't. That's okay. It doesn't really matter what it looks like. But it's something called teichoic acid, the green ones. And my analogy for this is always, if you've ever heard of steel-reinforced concrete, which is a building material. If we think of the peptidoglycan layer itself as sort of the concrete, these teichoic acids are sort of the steel rods, and what those sort of vertical elements do is to give a lot more strength to the concrete, in our little analogy, or in this case to the peptidoglycan layer. So if we think of the polymer sort of going left to right, these vertical elements, these teichoic acids, are going to reinforce the structure of this peptidoglycan layer and make it much stronger, which is, again, kind of the point, right? We want this cell wall to be very strong so that as water comes into the cell and the cell membrane sort of pushes up against it, we need that cell wall to be strong enough to not let the cell explode, right? So we need this to be as strong as possible. So that's what we think these teichoic acids are doing. And then a slightly modified version are these purple ones here that are representing lipoteichoic acid. So it's the same thing, it's teichoic acid, except it's got lipo, a lipid attached. And if you'll notice, that lipid allows the lipoteichoic acid to insert into the cell membrane. Okay, so this is the cell membrane here, here's the cell wall. So it sort of, in a way, kind of attaches the cell wall to the rest of the cell, which is, I suppose, a useful thing. And so, again, both of them are, again, sort of acting as these vertical reinforcing agents to strengthen the cell wall, and the lipoteichoic acid seems to have some role in keeping the cell wall attached to the cell. And then there's usually a small space between the cell wall and the cell membrane, and that's called the periplasmic space. So it's filled with fluids, it's filled with enzymes that help make the wall and things like that. So, but there usually is a tiny space in between the two layers. So that is sort of a standard cell wall. In this specific example, I use specifically a gram-positive cell wall for bacteria. So these bacteria, again, would stain purple when you looked at them under the microscope after you ran this gram stain. So if any of you take micro with me next time I teach it, gram staining is an incredibly important tool in microbiology. So despite the fact that it was invented in the 1880s, we still use it today. It is basically the basis for all microbiological taxonomy. So almost always the very first thing you want to know about your bacterial cell is it gram-positive or gram-negative? Okay? Because that's going to help you diagnose whatever it is and identify whatever it is and treat whatever it is. There's a correlation between different antibiotics and gram-positive versus gram-negative cell walls as well. So always want to know whether your critter from your patient sample is a gram-positive or a gram-negative type of cell. So again, this isn't just, nice little cell biological facts. This really does have clinical implications. Okay, how are we feeling about the basic structure of a gram-positive bacterial cell wall?
Would the cell being gram-positive or gram-negative affect the way they would treat?
It will, absolutely. There are some antibiotics that are better at killing gram-positive cells and some that are better at killing gram-negative. So yeah, that's another reason you want to know that. Okay, so that's one type. And then the other broad category, those that stain pink at the end of this process, are called gram-negative cells. And they have a slightly different setup for the cell wall. Not completely different, but a little bit different. And these are things like E. coli, that we hear about all the time, Salmonella, Shigella, what's another gram-negative? With the meningitis outbreak over in Princeton, Neisseria gonorrhoeae and Neisseria meningitidis are gram-negative cells as well. So there's a whole plethora of gram-negs out there as well. Okay, so let's kind of orient ourselves. So again, here's the cell membrane. Here's our periplasmic space. And here, much thinner, but here is our peptidoglycan layer. Even though it's thinner, it's set up in exactly the same way. So it's still this. It's still polymers of those two sugars alternating, the tetrapeptide tail off the NAMs, and cross links between them. It's exactly the same. It's just significantly thinner. So here's the thick gram-positive version. In a gram-negative cell, that layer is much, as you can see here, much, much thinner. Okay, but otherwise, the basic setup is exactly the same, okay, with the sugars and the protein part. Now, there is another difference in addition to the thinner nature of it, and I think it's because it's thinner. There is no teichoic acid or lipoteichoic acid in the gram-negative situation, probably because it's thinner, so it doesn't need as much structural reinforcement. Okay, but otherwise, it is just the thinner version, for the most part, of the gram-positive situation. The really big difference is this. There's a second, what looks like a second lipid bilayer, and it is. So it's kind of, in a sense, a second cell membrane, although we don't call it that because this is the cell membrane. So we call this membrane the outer membrane. So for a gram-negative cell, the cell wall is the thinner peptidoglycan layer and this outer membrane. So these two together represent the cell wall. So that's a pretty big difference. And for the most part, this is pretty much just your standard old lipid bilayer, just like we've been studying all semester, with one big notable molecule here. If you notice, if you look closely at this drawing, they've drawn the lipids in the outer leaflet, the outer part of the outer membrane, a little bit differently. Instead of circles, they're sort of more ovals, and instead of 2 lines, they have 3 lines, and then we have all these long lines coming off here. This is trying to represent a very unique type of membrane lipid called lipopolysaccharide, usually abbreviated LPS. And this really is the only place that you find this molecule. Is in the outer membrane, or the outer leaflet of the outer membrane of gram-negative cells. And again, if you're into structures, this is what it looks like. So it is multiple fatty acids linked to sugars, and then more sugar attached to it. So this blue part is representing this sort of, what looks like a sort of little hair-like thing here, is this. So that's a lot of sugar, and that's why it's lipopolysaccharide. It's got a lipid portion and a carbohydrate portion. And then here's the lipid portion here. So it's a rather unique kind of molecule. Like I said, this is the only place you find it. And its sort of claim to fame for us is that it is a type of molecule that's called an endotoxin. Endotoxin is something, there is a toxin, obviously, that is actually part of the structure of the cell itself, is what the endo part means. As opposed to an exotoxin, which is a toxin that's secreted from the cell. Okay, so this one is part of the cell wall itself, so we give it a special name or a different name called an endotoxin. So this is sort of a unique aspect to gram-negative bacteria in that they have this toxin that gram-positive ones don't. Okay, that's part of their structure. So sort of the interesting story is that while they're alive and this cell wall is intact, okay, it doesn't cause any problem. It's not toxic to us. The problem is when you have the infection, you go to the doctor, you get your antibiotic, you start taking your antibiotic, and you start killing off these things. When these cells die, they start basically breaking apart into little pieces. So it releases that LPS and it starts floating around your circulatory system. Then it starts causing problems. Not huge problems, but sort of the more generalized aches and pains, and it will give you a low-grade fever and stuff like that. So gram-negative cells sort of hit you twice. So they cause problems while they're alive, right? Obviously. And then they also cause you problems as you're getting rid of them. So it's sort of a double whammy with a gram-negative infection that you don't really have with a gram-positive situation. They only cause you problems when they're alive. Gram-negs can cause you problems as you're killing them off as well because of that LPS. So it's kind of a neat molecule that I'd like to give a little bit of detail on because, again, it's unique to gram-negative cell walls. Yes, ma'am.
Wait, so you said that when they're alive, does it cause a problem or it does cause a problem?
It does not, NOT.
And then when they're dead, that's when it causes a problem.
It does, because when they're dead, the cell starts breaking apart into little tiny pieces. It starts to disintegrate, releases the LPS, and that's when the problems begin. Yes, ma'am.
Just the gram-negative one, though?
It is.
Okay, the positive is bad also. Or it's not even
I don't understand.
You said that in gram-negative, it's when it's alive, it's okay, but when it's
Yeah.
dead.
Stop.
Okay, yeah.
Bacteria are alive and they're
Yeah.
infecting you, that's a problem, right?
Yeah.
Okay? So when they're all bacteria, when they're alive, if they're in the wrong place in you, that's a problem. That's the infection. Okay? No LPS, right? Never mentioned that here with gram-positives, right? Okay. So when you kill them, infection over. You're better. Gram-negatives, again, has this special molecule, this LPS, that when the cell is alive and it's stuck here, it's stuck in the membrane or stuck in the wall, it's not hurting you. When the cell dies, because you're taking your antibiotic, again, it starts breaking apart. I don't have anything to break apart into pieces. But it starts breaking up into pieces. Some of those pieces will contain LPS, this endotoxin, that starts circulating in your body and starts causing problems. So, double whammy. They cause problems when they are alive, as all bacteria do. And these, because of that LPS, give you those other symptoms as they die because they're releasing that toxin. Does that make sense? Okay, so those are the two major types of cell walls in bacteria. There are some other variations, but this is we're just sort of highlighting things here. So those are the two major types of bacterial cell walls. Okay, so ideally, I'd like you to be able to identify the components, of the different walls, so that you can distinguish between the two. As far as good, two types of bacterial cell walls. Make sense? Okay. The next type of organism that has a cell wall are fungi. So we'll look briefly at the fungal cell wall. It's going to be really simple because fungal cell walls look very, very, very similar to a gram-positive bacterial cell wall. Except they're going to have different sugars and different proteins. But the overall design, really, really similar. Okay, so here's the cell membrane of the fungal cell. Here is a periplasmic space. And then here is our cell wall, made up of sugars and proteins. So it's very, very similar to the overall design of a gram-positive bacterial cell. And actually, if you gram stain a fungal cell, it stains purple, just like a gram-positive bacterial cell does. So the structures are very, very similar. Now, instead of NAM and NAG as our sugars, fungal cells have two different sugars called chitin, which I think we actually studied briefly back in September. It's the same polysaccharide that's in the exoskeleton of insects. And another one, so those are sort of the darker lines here, and then the lighter lines here are a different polysaccharide called glucan. So instead of NAM and NAG, we have chitin and glucan. But pretty much the same story. We have these polymers of sugars that we then need to cross-link, right? Because again, just having straight lines lined up next to each other doesn't make you a solid. So we need to cross-link them. And the cross-linking is done by this third component here called manoprotein. So there's your protein, well, protein plus sugar, but here's your protein component as well. As you can sort of see, these sort of large ovals here representing the manoprotein sort of cross-link the glucans and the chitin polymers into a solid structure. So again, thematically, very, very similar to what we just saw for a gram-positive bacterial cell wall, but we just swap out sugar, swap out a different protein, but we get sort of the same effect. So chitin and glucan are the sugars, manoprotein is the protein component. All of that gets cross-linked into a nice, solid, strong cell wall for the same reason. Okay, so that this fungal cell doesn't explode as water rushes in, so that it precludes osmotic lysis. That's always the reason for the cell wall, no matter what it's made up of. So that one's kind of easy. Kind of make sense? Okay, so that one's kind of easy. Just swap out these three for those three, and you're kind of good to go.
I'm sorry, are they also going to have the teichoic acids in there?
Oh, no. So that is a difference. That's unique to bacteria. So those aren't there. So it's just those three components. Okay, so that's kind of quick. And then we get through this fairly quickly too, because we've already studied this. Already looked at the plant cell wall, at least components of the plant cell wall. So we studied this when we were back doing carbohydrates. So what's the major carbohydrate component? Cellulose. Right, and where are those? There's cellulose. Okay, so cellulose is going to be the major carbohydrate component. Right, so those are the sort of green tubes there, and we'll talk in a second about how we get them going in opposite directions. We'll do that in just a second. Okay, so we have cellulose is sort of the major carbohydrate component. Then we have this cross-linking glycan, so sort of the same thing here. Well, glucan, glycan, they're very similar kinds of carbohydrates. Okay, so they're going to help cross-link the cellulose. And then the protein component is this protein here called pectin. And also, as you can kind of see, kind of helps cross-link all the cellulose components as well. So again, once you know one really well, the others are really similar. You just have to keep straight which sugars and which proteins go with which type of cell wall. Okay, so we have NAM, and NAGs as the carbohydrates for bacteria, tetrapeptide tails as the protein part for bacteria. We have glucan and chitin as the sugars for fungi, along with manoprotein as the cross-linking. And here for plants, we have cellulose and glycan as the sugars, pectin as the protein that cross-links it all together. Okay, and this is the same picture I showed you back in September. Right, so we have all those. Remember, cellulose is a long polysaccharide of glucose molecules, but glucose held together by beta glycosidic bonds. Remember those, instead of being much more hinge-like, where you got a lot of rotation, these were much more sort of, or at least had a lot less rotation about them, which made them more straight lines. And then we use all of our hydrogen bonding capacity to hold the polymers together, making the sheet. And that's what makes it insoluble, right? Because there's no more hydrogen bonds left to form with water, so that made it insoluble. And then this last slide is kind of neat. If you look closely here, each layer of cellulose goes at a 90-degree angle to the layer above and below it. So if it's going straight out at you, the next layer will be going left to right, and the next one will be coming out at you, the next one will be going left to right. So the question is, or a question is, how does that happen? How does the plant cell orient the cellulose in the sort of two different directions? And that's shown in this last slide here. In the membrane is the enzyme complex that makes cellulose. So that's cellulose synthase. And it synthesizes cellulose. It makes cellulose. And interestingly, those are very, very closely associated with microtubules. Okay, one of the cytoskeletal elements we've been studying for the past couple days. And most likely by means of motor proteins, these cellulose synthase complexes are sort of walked along these microtubule highways. Okay, we're used to seeing that. And off comes the cellulose in one direction. And then once that layer is done, the microtubules are reoriented. So instead of going this way, they'd be going left to right. Now the cellulose synthase complex would be walking along left to right, and the next layer under that would be going this way. Then they reorient again, and the next layer would be just like this one, going top to bottom. Then the microtubules reorient again left to right. So it's kind of a clever mechanism. Now, how exactly it knows exactly when to do that and how the microtubules are organized to, kind of be going up and down versus left to right is still, I don't think, completely understood. But this seems to be at least part of the story for how the different layers of cellulose get laid down at 90-degree angles to each other. So it's kind of a neat story that links us back to microtubule highway systems and motor proteins and enzyme complexes. So brings together quite a number of the stories that we've been looking at recently. Okay, so that's a little bit about cell walls. Does it seem okay? So I would like I said, I would like you to be able to know which carbohydrate and which protein goes with which kind of cell wall, basically, is the major thing I'd like you to know there. Does that make sense? Okay. Animal cells have a layer outside themselves as well. That's usually referred to as the extracellular matrix. So it is sort of equivalent in that there's a layer outside the cell, but the extracellular matrix is nowhere near structurally as strong as a cell wall. So this extracellular matrix does not keep the cell from exploding during osmotic lysis. Right? If you remember, animal cells solve that problem not with cell walls, but with an extracellular fluid. Our body fluids have the same concentration as our cytoplasm does, so they're isotonic. Right? So the extracellular matrix here, while it's a layer outside the cell itself, is not a cell wall, so it's not keeping it from osmotic lysis. We protect our cells from osmotic lysis by setting up an isotonic body fluid. Instead, what the extracellular matrix in animals does is to essentially help keep cells together in a tissue. So why do all your liver cells stay together in your liver? How come they don't start floating around your abdominal cavity? And why does your kidney cells all stay together in the kidney and not start floating around? Partly, the answer is this extracellular matrix. It is sort of like a glue, if you will, that keeps cells together within a tissue. It's not the only reason, but it certainly helps keep cells together in the tissue. The other big answer to that question will be the junctional complexes that we're going to look at as sort of the third piece of this topic. But I like to do the extracellular matrix right after cell walls because there is that general similarity in that it's an extracellular layer. So there are a number of different kinds of extracellular matrix. So our hard bones are a form of extracellular matrix. Right? These are going to help keep those cells together in sort of a mineralized, really dense, hard kind of situation. But cartilage and other kinds of connective tissue are also forms of extracellular matrices that, again, help keep cells together and certainly, obviously, do other functions as well. The bones help, give our body shape, and cartilage helps to cushion joints and things like that. So there are certainly other functions, but we're going to sort of look at them generally as a way to keep cells together in tissues. So if you take Dr. Ingersoll's histology class, you'll learn much, much more about the different kinds of cells that make the extracellular matrix, like fibroblasts and things like that. We're just going to focus on some of the components of this system. Okay, so we're going to look at a number of different kinds of proteins, just real briefly, that make up the extracellular matrix. So things like collagens, elastins, proteoglycans, which we've already talked about. I think we mentioned collagen way back. Other things called fibronectins and laminins. So there's going to be a number of proteins that go into making up the extracellular matrix. So collagen, I think we had looked at when we were looking at protein structure in general. This was a really good example of a protein having really just one major type of secondary structure. Remember, this was sort of one long alpha helix, which was an extreme example. But it was also a good quaternary structure example because 3 individual collagen proteins wrap around each other into sort of a triple helical kind of shape. And then those triple helices link up with other triple helices to make thicker and thicker collagen fibrils first, and then fibrils get together to make fibers, so we keep kind of ratcheting up the structure here. But eventually they can make fairly thick structures called collagen fibers that are obviously very, very clearly seen under the electron microscope. Collagen is a huge percentage of animal cells' extracellular matrix, ECM. So this gives you another view of how this is put together. So here's the single chain. 3 of them get together into a triple helix. A bunch of those get together into a fibril. A bunch of fibrils get together to make a fiber. So again, it keeps getting thicker and thicker as we do it. And there are a number of different kinds of collagens, as you can see here, that are typically tissue-specific. So there looks like at least 19 different collagens have been identified. And they're all just slightly different varieties. Sort of the same idea that we saw the other day with the various kinds of intermediate filaments. There were different classes of those. We have different collagen types here as well. So once we kind of build the larger picture, you're going to see many, many, many collagen fibers as part of the extracellular matrix. Another protein that you'll see in a decent amount is called elastin. And I assume it was named elastin because it has certain elastic kinds of properties. So it kind of can stretch and relax. Okay, so that could be a good idea, especially as you move your arms and things and you change angles and stuff. It would be good if some component of this had some sort of bendable, elastic kind of characteristic to it. And that's what elastin kind of brings to the picture. But you can also see that these are cross-linked as well. So individual elastins can interact with each other to, again, help form a more solid kind of structure. But this one, again, has more elastic sort of properties to it. And then I think we had looked at proteoglycans when we were doing carbohydrates as well. Remember how this works? The prefix is the smaller amount, and then the main word is the one with the larger amount. So proteoglycan has a little bit of protein with a lot of sugar. Okay? And these are good examples. So typically, we have kind of like a small peptide to which is attached just a huge amount of sugar in these kind of situations. And again, we're not going to get into lots of detail here, but just remember the general structure of these proteoglycans. And again, these are going to be important components to the ECM. In certain kinds of tissues, especially in your joints, these are also very useful because with all the sugar and all those hydroxyl groups and ability to form hydrogen bonds, these usually bring a lot of water into the mix. And when you mix water with these, it sort of makes kind of a jelly-like viscous fluid, which also helps lubricate your joints. I think I mentioned this is all the fluid in your eyeball, or proteoglycans dissolved in water, basically. And so these can also have lubrication types of functions as well. And then the next couple of proteins are really useful to start cross-linking some of these all these other components. So this one's called fibronectin, and you can see that it has binding sites for other things like collagen, like fibrin and heparin, and I think we'll look at in a little bit. And it can bind to cell surface receptors in the cell. So this is very, very useful for other things attaching to it, and we're starting to build sort of this larger structure. And these are just stained cells here's actin, so we've seen that before. Here's cell stained for fibronectin, so there's lots and lots of fibronectin around. That's fine. Okay. In epithelial sheets especially, so think your intestinal cells, that's probably a good example, which I think these are. Okay, so here's these are either your ciliated epithelial cells in your trachea, or these are the villi in your intestinal cells. It doesn't really matter. But the sort of bottom layers, so you can see all these epithelial cells are sort of cemented here to this connective tissue here by means of this layer is called the basal lamina, so sort of the basement layer, if you will. So you tend to see a lot of extracellular matrix here that kind of glues all of these epithelial cells to this connective tissue here, that in this example is being secreted by these fibroblast cells. And in the basal lamina, you find lots of this type of protein called laminins. Now, be careful. We've seen a related word before, right? We've seen lamins, which were those intermediate filaments inside the nucleus, right? These are laminins, two ins. Laminins. Okay, so be careful, lamins versus laminins. Okay. So these are, again, components of the extracellular matrix, usually, again, found in this basement layer, this basal lamina. But, again, have binding sites for other things. So, again, here's a collagen binding site. It can bind to cell proteins in the cell membrane of the cell. What else? Again, this heparin binding sites. So again, these are good cross-linkers of other pieces of this extracellular matrix. And the receptors in the cell that a lot of these things are going to hook up to are these proteins here called integrins. We're going to study these more on today's Wednesday, Friday, when we do junctional complexes. But just to point them out here, where it says, cell surface receptor binding domain, this typically is the type of receptor they're talking about are often these integrin proteins. Again, more on those on Friday. And this is kind of what we're going to look at on Friday. So here's the extracellular matrix all cross-linked. Here are the integrins in the cell membrane. So it cross-links the cell to the extracellular matrix. And then on the outside, then on the inside, we'll see it's going to cross-link to various components of the cytoskeleton. Okay, so we're going to make connections between the stuff outside the cell, across the membrane, to the cytoskeleton on the inside. But we'll do more of that on Friday. So here's sort of the overall picture. So here's the cell membrane, here's the cytoskeleton stuff that we'll do on Friday, and then here's all of these other proteins that we've been sort of talking about. So here's the integrins, here's fibronectin, here are collagen fibers, here are proteoglycans, all sort of cross-linked together to create this sort of layer, this glue layer, if you will, on the outside of these cells, helping to keep them together in a tissue. It's sort of the major claim to fame. So what I'd like you to be able to do is recognize this sort of short list of proteins as components of the extracellular matrix. Okay? Okay, so we'll link this